Hello, and welcome to the Musella Foundation Brain Tumor Awareness Month webinar series. Tonight's special guest is Dr. Bert Nabors. Dr. Nabors is a distinguished professor of neurology, vice chair for clinical research, and director of the Division of Neuro-Oncology at the University of Alabama at Birmingham. We're honored to have you with us, Dr. Nabors. I will turn things over to you.
Great. It's exciting to be here. And this is actually a new talk that I've kind of pieced together. I did this when Dr. Musella asked if I would speak, and it kind of made me go back and work through some things that are of concern to me related to the field, particularly glioblastoma. I've been in practice here at Birmingham for 30 years, and 28 of that has been dedicated to neuro-oncology. And so I want to share some thoughts. And so let's get going. A few just kind of intro slides before I give you my little outline. So, oop, hang on. Okay. So we know the impact of glioblastoma. The median age is usually for those in trials in the 50s and then overall 65. Standard treatment revolves around surgery, radiation, and chemotherapy. Unfortunately, the older we are, the poorer we do. When we look at the burden of this disease on patients, that's measured by the NCI in a metric called years of life lost. It's high, amongst the highest, both in the UK and in the US. In addition, it carries a pretty tremendous public health cost. This is distributed not only to the payers, but also to the patients and their families. As opposed to a lot of cancers that we are aware of, this one truly spans all the way across life. It is in children, which we'll describe as under 14, the adolescent and young adult, which is the AYA population, so those 15 to 40, and then older. Uh, Unfortunately, when we look at both children and the AYA population, which this is getting up to people 40, it's one of the leading causes as far as incidence of cancer and one of the leading causes of cancer death. As obviously we get older, we begin to have other types of cancer, such as prostate, breast, lung, colorectal. It falls down. But I think this is an under-recognized burden is particularly this weight on children and young adults. We're all familiar with this classification. It gives us what the survival is for patients across the different disease types. So astrocytoma versus oligodendroglioma versus glioblastoma, and then using the WHO's classification from 2021, adding in the IDH status. So we can see from these survival curves, actually, there are times we may be doing pretty well, but unfortunately, substantial times we're not doing very well at all. And so this is what I wanted to talk to everyone about today. So there's three objectives. Some of this is probably my thoughts or the way that I'm piecing together results from the literature or maybe my own experience. But I view three things that I want to touch base to everybody with. So one, why do people get glioblastoma? Two, what is our goal? Your brain or the glioblastoma? And then I want to go back to an editorial I wrote a couple of years ago. And so this last section is entitled Back on the Road We Travel. All right, why do people get glioblastoma? So, diseases occur basically because of two major reasons. It's either something about our nature or our nurture. We'll consider nature our genes and nurture the environment. So on the gene side, there is an effort that I'll touch base in a little more detail relative to a type of analysis that a few groups across our country have really led called genome-wide association studies, or GWAS. Come back to that in just a second. Very important efforts, though, involving thousands of patients. There are familial syndromes where people have a predisposition. So Life, Remini, Turcotte, and even neurofibromatosis type 1 have a high risk of, or higher risk, elevated risk of a glioma. There are often scenarios where we see multiple family members with a glioma. Certainly, this would speak to something that is gene related. And I also put over here, and this is work that Melissa Bondi at Stanford and Quinn Ostrom at Duke have really led, and I think we need to kind of be thinking of this. There seems to be somewhat of a protective effect for people with autoimmune disorders. So if your immune system's on a heightened sense of surveillance, there may be a protective effect. When we look at our environment, there's certainly case cohorts. So those cohorts are, the recent report maybe a year ago about the New Jersey high school where a lot of alumni had developed brain tumors across a wide spectrum, meningioma to glioma. Been some concern about a specific Major League Baseball, maybe field or stadium, in which a number of people who I think were primarily pitchers might have developed glioma, along with Jill Barnhart Sloan and others, Brett Thieler at Walter Reed National Medical Center. We have had this interest of looking at associations with military service. And then I would reference this book for all who are really interested in something to read. It's about Tom's River. The title of the book is Tom's River, A Story of Science and Salvation. The author is Dan Fagan. Really quite an interesting story, particularly ties into pediatric development of brain cancer, but worth reading. As far as lifestyle, is there something that we are really doing, such as alcohol use, smoking, cell phone use? Really, I would kind of say I don't think we see much of an association in that particular space. I think another aspect, and I'm mainly interested, this is from the CB Trust report, and this was October 2024. I don't know if you guys can see my mouse, but I'm circling the top part, which is A. These are the malignant tumors. And it's just a map of the United States showing the average annual age-adjusted incidence rates. And really, there's not a ton of difference here. We don't see a whole lot of change across our country, suggesting there's a heightened area of worsening in one state or region as opposed to another. I took a look, and this has been something that I find quite interesting. Um, this is an update to the SEER data, but it's an observation that, is known. So if we look to the left, this is again the age-adjusted incidence rates. The left over here, which are the triangles, the green triangles and the blue triangles. This is just dividing by male versus female. So the top is male, the bottom is female. And so by sex, there's an increased risk or increased incidence if you're male. The right side, where we have this empty circles on the top, these are whites, and then the brown boxes on the bottom are black. I couldn't figure out how to make the seer allow me to put white men versus black women. But there's a dramatic difference in what I'll think of as incidence, but that must tie into risk. So it suggests that despite uh, 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 our biological and molecular understanding, there must be a protective effect tied to being a black woman that we don't know. It's just the opposite for meningioma. Now, I told you I would talk about GWAS a little bit. These are genome-wide association studies, and this will definitely be a little bit because it's not an area that is my expertise. The GY study stands for Genome-Wide Association. There are the only really recognized environmental risk factor for glioma is exposure to ionizing radiation. This has typically occurred in children, either for the treatment of tinea capitis, which is ringworm of the scalp, or back when craniospinal radiation was being deployed to largely cure children with acute lymphocytic leukemia that had infiltrated into the nervous system. Um, there is evidence for a genetic risk, though, and that's tied to there is an elevated risk of brain tumors, glioma, if you have a first-degree relative with a glioma. And then, as I mentioned earlier, there are a number of inherited conditions for which there is a predisposition. I think a total of 3 national to international GWASs have been done. They've identified 27 glioma risk SNPs, which stands for single nucleotide polymorphism. And these are just variations in the normal nucleotide sequence of genes for which there is a heightened risk for the development of this cancer. The next goals relative to this work are to try to understand how specific risk SNPs of these 27 are associated with particular molecular subgroups, or what's the functional implication of this? What does this variation do that alters a function that could be implicated in the formation of the disease? So still important work and a lot more to actually do. Now, one of the observations or one of the things that's always interested me, so I do think, there is a component of an environmental exposure, but maybe that's a natural event. I mean, it's not something that's man-made or a toxin or a pollutant. Maybe it's something that occurs naturally, particularly since the incident rate over decades seems to be so unchanged. We looked at our numbers of at least glioblastoma cases, and this is the average cases we see each month for every month of the year. So you can see there's a couple of months, and there's no statistical analysis. This is just me, looking, exploring for things for which we may ask questions. But there are a couple of months, May and June, for which the number of cases we see is a lot greater or is larger or more than other months. I think we have to remove December. A lot of clinics get closed, but we're averaging, 65, 70 new GBM a month. In those months, we're, probably the other rest of the months we're closer to an average of 50. Suggests that there must be something tied to exposure and maybe something natural. Another aspect that we're involved in locally is trying to better understand exposure related to the watershed in Alabama that you live in. So the United States Geological Survey has a method where they classify watersheds, and they can break them down, pretty specifically. And these are called hydraulic unit codes. And the little graph here to the right simply, oops, let me try to go back here, is simply trying to show at a, high level and how we can, go from, what may be the 2-digit code, which could be this region of California, 4-digit, which becomes a subset of that, 6, 8, 10, down to 12-digit, where we're getting down into smaller specific water supply areas. And so our interest is using this across our state. So these are defined with the acronym HUC. There are 4 major watersheds across our state. We keep up with the age-adjusted incidence rates in our state. And when we look at these across the watershed regions, we can see that there is a difference. Again, this is crude, and it's burdened by the fact that Alabama has many, many rural counties, and many of these areas tied to the watersheds harbor our more rural areas. So there're just not a lot of people. So we just don't end up seeing a lot of cases from these counties. And then our ability to be sure we captured everybody can be a challenge too. But we can see that there is a difference, from maybe a very low rate to a higher rate amongst some of our watersheds. I think this is important as we try to better understand, what is driving the cause of this disease. So to finish kind of my little first section, key points, etiology of glioblastoma is not known. The SEER data shows pretty stable incidence, and this has been over decades. I think we can think that the likely cause is a background of genetic susceptibility. The GWAS is telling us that, but in the right environment, the right environmental exposure. Need to better understand what those particular triggers are. I think also important for us to remember there is a suggestion of an ability of the immune system to prevent or reduce the risk of the disease. And this has been largely observed in people with autoimmune disorders. All right, the next piece I wanted to talk about, and this is, my section about the goal, and the goal being specifically your brain or the tumor. And, the rationale behind that, and something that I'm always telling patients, is unfortunately for glioblastoma, we cannot cure this. The goal of our treatment needs to be to preserve neurological function, and obviously we want people to live longer. All right. So when we think about the brain, it is the ability of a human to do whatever he or she wants, needs, or desires, and it's driven solely by the nervous system. We can divide the nervous system into the central nervous system and the peripheral. The peripheral has malignancies that occur to it. We're not going to include it. We're mainly going to focus on the central. The central is the brain and the spinal cord. And I'm really predominantly interested in the brain. The brain is divided so that different functions reside predominantly in certain areas. An example to look at may be speech. If you are a right-handed person, then the predominant localization of where your speech function is in the left, and it's in a region called the temporal lobe. But different functions, as is illustrated on this schematic, are in different regions of the brain. Even just this predominant function is important as opposed to another tissue for which that whole tissue is doing the same function. An example may be the pulmonary system or the lung, which is important in the oxygenation of blood, but the right side is doing the same as the left, the top is doing the same as the middle as the bottom. Very different in the nervous system. So there is an importance to function tied to specific locations. The next level we're concerned with is with a couple of primary cells of the nervous system. So the neuron is the one we simply think of as a cell involved in the performance or the execution of some neurological task or activity. This may be like motor or sensory, it may be vision, smell, taste. Could be something more complex, such as memory, concentration, attention, or could be very complex, like abstract thought. So the neurons are the largest drivers of these functions or activities. The glial cells from which glioma are derived and which are a essential cell type within the nervous system. The nervous system does not function or exist without the glial cells. But they are not the drivers of these specific functions as I'm trying to share. The neurons accomplish almost all of these tasks by the formation and modulation of the synapse. Okay, this is the little region in which it forms a chemical connection with other neurons and can generate an electrical event which may affect one cell or may affect lots of cells. We'll say millions of cells. And this is something that's highly modulated. And that modulation helps form our ability to do things that we don't really think about anymore. And a simple example may be in the morning when you're brushing your teeth, you identify your toothbrush, you know what to do with it, and you do that. A lot of times where people may have an impairment to the region of the brains important in visual spatial orientation, the recognition of a complex geometry, that may require a lot of conscious energy for them to accomplish. And this is what is occurring with a lot of our patients. There's another level of connection that the neuroscientists are extremely engaged in, where areas influence a far-ranging aspect of our brain. This is called the connectome, and this is a illustration of that. I mean, I think it shows you how massively connected the central nervous system is and how functions that you may not necessarily think would be disrupted by a tumor in a certain location may be driving some complex issues for patients and their families. Okay, along with this, when we think there's two aspects that I want to kind of talk about. One, is the impact of the disease on the nervous system, and then briefly, really just kind of listing, but the other is the impact of the treatment on the nervous system. So disease-wise, probably the biggest driver is this location. So outcomes vary depending on where in the brain tumors are. There's a number of groups that have looked at this. Locations where things may go not so good, or central locations. So this is the corpus callosum. This is a white matter tract that's connecting both hemispheres together. Obviously, through that is information that is allowing the right side of your brain to interact with the left side of the brain. Basal ganglia, which is a complex system for providing fluidity of motion, or any regions that are near the third ventricle, which is deep brain, hypothalamus, basal ganglia, thalamus, and then the parietal lobes. Parietal lobes, I think, are often uh, maybe not as appreciated as having such an impact on a patient. The parietal lobes have a lot to do with visual spatial awareness, and that can be very disruptive to function, to being able to do what you want to do, or being able to do your day-to-day activities. The best locations are on the right side, temporal or frontal lobe, and then the further away from the third ventricle, the better. I really never developed this slide very well, and maybe in future versions I can get it better, but there clearly is a big impact of treatment on the nervous system. Some of this is neurological, some of it is higher neurocognitive function. Obviously, there's impacts from an emotional perspective, particularly when we begin to look at one of the most common medicines we use, which is dexamethasone. And then also mental health is probably often overlooked, as we spend time trying to sort through what to do disease-wise. I want to share one slide of some of the work that's going on in my lab. This is with my colleague, Natalia Filipova, who largely runs our lab. We're very interested in mediators of this neurological change. Obviously, we're having to do this at the cell culture animal model level, but we're particularly interested in a molecule that is secreted by myeloid cells or glioma cells. It's called TRIM. It stands for triggering receptors expressed on myeloid cells. And then the receptor, one of them, there's a whole network of these, is called DAT12. It's got some other names, but this is activated by TRIM1. It drives the expression of complement and the activation of that cascade, particularly an effector molecule called C1q. And so there's a lot of pathology associated with this. It's not specific for glioma. We believe it applies to glioma and that it is partly responsible for the neurocognitive impairment of patients. If we look at the expression of this effector that's driven by this DAP12 pathway, we can, which is called, it's just, we're calling it C1q. We can see it's relatively low in normal brain. This has gotten most attention in neurodegenerative disorders. So this is Alzheimer's disease or Huntington's, but actually glioblastoma, it's multiple folds higher than in these other neurodegenerative diseases. It works by creating neuroinflammation. It leads to demyelination on axons. I'm sorry, we misspelled synapse, but it leads to disruption of synaptic modulation and the manipulation of synapses, which are essential for the formation of memory. Short term, meaning like what I'm going to the grocery store for, versus long term. And a lot of that has to do with disruptions to a process where proteins that interact with RNA alter their expression at the level of protein. So a lot of exciting work going on in our lab. We've got a grant we're putting in June the 5th, where we're trying to further develop some lead molecules that Natalia has identified that we believe have some degree of potential clinical applicability. So neuroprotection, I'm often asked, well, what can I, as a patient or a family member, do? The number one thing that you can do that is protective or restorative to the nervous system is activity. That is the primary driver of neurological improvement and outcome. This is for any neurological condition there is. If it's a static event, like you had head trauma or you had a stroke, it's the same as if it's a dynamic process, like you have an inflammatory condition, like a demyelinating disorder, viral encephalitis, or a glioblastoma. The only thing that restores neurological function is activity. There are supplements that I think make sense. The B vitamins with folate, all are essential for the function of the nervous system. I don't really believe you can get too much of the B vitamins, but I only would recommend taking what's recommended. If you have a problem with absorption, particularly of B12, which really should only occur in people with pernicious anemia, then you may need the injectable version. And I think vitamin D3. Diet, often ask about this. At the very end, I got just a comment on the ketogenic diet, which I support. But I think diet in general, and I'm not a dietitian, but I think for most Americans, this is often a big challenge, and making major dietary changes is a big challenge. I favor, looking at a diet that's more anti-inflammatory in nature. This means high in foods that are antioxidants. These are usually fruits and vegetables, and the purpler the fruit or vegetable, the higher the antioxidant content. I think we have to think about the treatments when we're presented with these options. Surgery has advanced tremendously. There's obviously quite sophisticated capacity to either see with fluorescent dyes, to image with intraoperative MRI, or for the surgeons to map out function after awakening a patient in the operating room. These surgeons have to be sought out. Also, I think the literature is very strong for those with significant neurological impairment or older patients. And that older was 65. I'm 62, so I don't really like thinking about that as older, but a reduced dose of course. So from 42 treatments to 21, so over a 3 week period, 15 over a 3 week period, and from close to 60 gray to a 40 gray. So, and you have to incorporate how old you are and what your goal is, because the older we are, disease-wise, we just do worse, but we just don't tolerate the treatment as well either. So the key points on this section where I'm talking about the goal is your brain or the glioblastoma. Our primary goal is preserve neurological function. Neurological function extends beyond specific locations in the brain, and improved understanding of how glioblastoma and treatment drive neuroinflammation is one aspect of trying to do better with our current treatments. Okay, back on the road we travel. This is the last section. This is an editorial I was asked to write back in 2023 by Neuro-Oncology, and it was in response to a large randomized phase 3 blinded of the addition of PD-1 inhibitor to newly diagnosed patients. So some people got temozolomide radiation, some got temozolomide radiation, nivolumab. I think it was blinded. I may have misspoke there. But unfortunately, the study was not favorable. It showed no benefit to doing this. And it really had me thinking through, our course. And so one of the few things that I learned in high school English was in 10th grade, we read the little book, Death Be Not Proud by John Gunter, who wrote this book back in the I think 20s or 30s. He was a travel correspondent for Time Life. His son, who was a college student, developed a glioblastoma, probably was an astrocytoma, WHO4 IDH mutated back then. And he chronicles his uh, uh, course and his treatment and the distress on the family, which is quite similar to what it is today. His survival was 18 months, which, not all that off from today. Actually, I can see in my little thing, this book was published in 1949. So we're, now decades since and probably doing about the same. It occurred, this editorial I wrote occurred following an ASCO, American Society of Clinical Oncology, in which this small investigator-initiated study was reported for patients with advanced colorectal or rectal cancer, who all enrolled on this study with this immune checkpoint inhibitor. They were biomarker selected, so it's just not like anybody. There was a specific reason they went on, but all had a complete response and never went on to get the standard of care. And so, struck me as, a standard of care that actually works in that disease, meaning surgery, radiation, and chemotherapy, but probably with a good bit of morbidity tied to it for the patient. And it just struck me that, we're a little bit too tied to kind of what we do. Okay. And so let's look at this. So imaging, we often see, these types of enhancing, masses somebody presents all out of the blue. They usually don't have anything else wrong with them. And are suddenly seeing somebody like me talking about something that is called a glioblastoma. We can see on this image there's a mass, there's mass effect. The ventricle, is a face. There's this dark signal, which is bright over here, which is edema. The neurosurgeon has operated. They've removed all of that. There's some restoration of that mass effect. The neuropathologists get the tissue. They will see too many cells. The cells can have mitotic figures in them. There may be necrosis, or if they do a stain of how many cells are going through the cell cycle, it may be real high. And so even by the 2016 criteria, this would be called a glioblastoma WHO4. we would talk about radiation therapy, concurrent radiation with temozolomide, probably go over what the clinical trial landscape looks like, talk about other things like what steroids are for, what are their side effects, if you need to be on an anticonvulsant or not, and then prognosis. And so, this current treatment is all from work reported back in 2005 by the EORTC led by Roger Stoop and Monica Higi. This top survival curve is everybody. So obviously, they did better, when you got chemotherapy with the radiation than with the radiation alone. But we're still, when we look at the number of people and we're kind of out at 2 years, it's a small number that are actually making it, even though these survival curves are separated. If we then look at this molecule, which can predict if you are sensitive or resistant to chemotherapy with temozolomide, so the expression of this gene called MGMT methylguanine methyltransferase, if it's not expressed, the name of that is unmethylated MGMT promoter versus methylated. We do begin to see what I would describe as a more meaningful separation of the survival curves. But when we kind of look at the numbers, it remains, a very small number of patients that can escape. So we've kind of got a tail that's adding this separation. And then around 2023 or 22, we added tumor treating fields, which improved that, a little bit more. Uh, one of my responsibilities is I've been the chair of the National Comprehensive Cancer Network's CNS Tumor Guideline Committee. Dr. Musella, often submitting recommendations. And one of the things, that if we just, and the purpose of these guidelines is to make sure that our community-based medical oncologists, radiation oncologists, no matter where they are in our country, and we have even covered a lot of other countries, significant portions of sub-Saharan Africa, Poland, working into South America, but that they can have their guidelines look similar to ours. But unfortunately, we really, even if we know your tumor may not be responsive, because of the clinical trial landscape and what's available, we're still providing a higher recommendation of using a drug that may not actually be that beneficial to a patient. And so, what is it that we are really kind of missing related to this disease? There are many critical phenotypic features of glioblastoma that are drivers of bad behavior and ultimately lead to the loss of function for patients. Obviously, this proliferation capacity within the enclosed nature of the skull. This process of tumor-associated blood vessel formation called angiogenesis, I think what's kind of missed here, it's not just this issue of these tumor blood vessels kind of helping the tumor grow, but the disruption, these tumor blood vessels, tumor-associated blood vessels cannot form an intact blood-brain barrier. And so you get extravasation of fluid way outside of the area where there's predominant tumor or where tumor is infiltrated. This alters the ability of the brain to auto-regulate blood flow. So the brain, your nervous system changes where the blood is flowing to in your brain based on what you're doing or what you want your brain to be doing. And so more blood flow going to the temporal lobe if you're taking a test and you've got to remember stuff versus if you're running a marathon where probably more blood flow is going to the motor cortex. And so this creates regional ischemia. And so you're like somebody who's had a stroke, in this case, almost a quarter of the brain versus what would look like a relatively small, tumor. These tumors also invade. This invasion is akin to metastasis in systemic solid cancer as a big driver of morbidity and mortality. They rarely leave the nervous system, but they certainly invade. And then they are very resistant. They are extremely immunosuppressive. They create an environment in that tumor area where they suppress and actively keep the immune system out. Now, a lot of work through the TCGA and the Human Genome Atlas have allowed us to identify critical regulators of these signaling cascades driving the proliferation, driving the angiogenesis, and to a certain degree has probably led to some specific therapies. And so when I summarize this section, my key points on glioblastoma, all right, we all know that glioblastoma WHO4 and astrocytoma WHO4 are not the same entity anymore. Astrocytoma is IDH mutated. MGMT promoter is unmethylated, and there really is a limited benefit to any temozolomide. We got to be moving into a space where we accept that, and we're going to do clinical trials where it's left out, it's eliminated. Next generation sequencing, which is the molecular analysis, is important to generate additional diagnostic information. So there's a number of new subtypes. Prognostic, that's important to be able to share with patients and families, as well as therapy. So there can be alterations in specific genes for which they are altered in other cancers for which a drug has been developed. An example would be something called a fibroblast growth factor receptor, FGFR. 3 is the usual one, often fused to something called TAC3 in glioblastoma. There is an NCI clinical trial providing access through a drug called erdafitinib. And if you can't get on the clinical trial, we try to prescribe it off label. Yes, insurance will deny it. We'll appeal that, but then we would submit to the manufacturer. And so we may create therapy indications. And then obviously there's a need for innovative clinical trial designs. We've got to be more creative. So I want to lastly share with you, related to glioblastoma, some of the work that we've been doing here in Birmingham. And so this is results of a study that was reported at SNO. It's called INB 200. Was a phase 1 study of gene-modified autologous, that means they came from the patient, gamma delta T cells. So this is a specific type of T cell that has a very innate capacity. It's like a natural killer cell in that it doesn't have to adapt. It doesn't have to learn that that is cancer. It will attack without having to learn. And this was for newly diagnosed patients with GBM and that were planned for standard treatment. So there were 3 dose cohorts. Everybody got the same dose of these cells, but patients got different infusions of the cells. So first group got 1, next group 3, last group got 6. The way this would work is the patient had to be consented. They had to agree to be in the study, or at least the first part, which was to have an access device placed into the tumor before they even knew what they had. Now, granted, a lot of times we can be pretty confident based on the imaging that there's a high likelihood of this being a glioblastoma. They would undergo a surgical resection, have this access device, which we call the RICM, placed, and then they would undergo apheresis. So they would have gamma delta T cells collected. While they were doing radiation and Temodar, the gamma delta T cells were being altered to make them resistant to temozolomide. So they were being transfected with the gene MGMT to make them resistant. You don't want your lymphocytes all killed by the drug you're taking to try to kill the tumor. And then they were being expanded. And then once patients entered the maintenance phase of chemotherapy with temozolomide, on day 1, they would get an IV infusion of temozolomide. and then I would inject the gamma delta T cells, 10 million at a time, back into the surgical resection cavity. Obviously, as a phase 1 study, the goal was to make sure this was safe and try to see if we ran into any toxicities by giving this more than once. The rationale behind this is we're trying to target the DNA damage response through a combination of both cell-based therapy and chemotherapy. Interestingly, temozolomide, even if you are MGMT resistant or unmethylated, will induce the expression of the antigen, that the cell surface receptor that the gamma delta T cell is trying to hone to, which is called NKG2D. And so giving Temodar, the glioma cells are expressing this on their surface so the gamma delta T cells can find them. So even if resistant, there was a potential benefit for these patients. Here's the swimmer's plot from the study. It's showing survival. This particular patient that's way down here was actually the only one in the study who had an IDH mutated study. It had an astrocytoma WHO4. When the study began, it was called a glioblastoma, IDH mutated. That wasn't an exclusion. Doesn't need to be really for a phase 1 type of study. This pink bar here, column, is actually the median progression-free survival of the Stoop regimen, which I was showing you earlier. Here's an example of a patient, and we can see a series of slides through the course of the disease. This patient ended up completing cohort 3, which was 6 infusions, intracranial injections, did progress. Tumor tissue was collected. At diagnosis in the original tumor, there were very few effector T cells, CD3 or CD8. But then at recurrence, so at relapse, there was a heightened infiltration of CD3 and CD8 cells, which we want to believe is somewhat related to the effect of the gamma delta T cells. Now, another area, and this is getting close to my last bit, so just hang in with me, is really some exciting work going on by one of our becoming more mid-level faculty, Braden McFarland. And this is really about the gut-brain axis and how this may impact disease. And so, people hear about this a lot. I'm not going to go into all the signaling molecules that may be derived from the GI tract, but they have a substantial impact on aspects of central nervous system function. Now, we do real well with anti-PD-1 therapy in mouse models of glioblastoma, but as we talked about earlier, we haven't done so well in humans. We don't see that same effect. Most preclinical studies in glioblastoma are in mice, and they're in mice that have a mouse microbiota. So the microbiome in the gut of the mouse is a mouse's, it's not a human's. And there's big differences between a mouse's and a human. They're really dramatically different. Up to 85% of the gut bacteria that are in a laboratory mouse GI tract aren't in a human. Okay, so they're very different. Dr. McFarland generated these mice with humanized microbiomes. Okay, so she took donor feces, isolated the microbiome, and created these in a type of mouse called a notobiotic mouse, which has no microbiome. So it's transplanted with fecal samples from human donors and then creates a number of lines. They're called humanized microbiome. There's 5 of them. 5 colonies, numbers 1 through 5. Now, interestingly, if she looks, and I'm going to only focus on 2 this end for the different strains, the different mice, there are 5 of them. We can see they have, their microbiomes cluster nicely together, depending on that strain. And if we look at in this just bar graph, the expression of different types of microbiota, you can see it's obviously very different, as it is amongst all of us. Now, interestingly, when she uses some of these models, implants glioblastoma, and then treats them with PD-1, there are some that don't respond, behave like we see in human clinical trials, but then there are others that actually respond, where we see an effect of the PD-1 inhibitor. And if this group are treated with antibiotics, that effect goes away. So it suggests that there is a role for the microbiome in setting a stage for sensitivity. She and her lab team are working out the cells that are involved in the signaling pathways that are involved in that. I'm often asked about complementary therapy. I've talked to you a little bit about supplements. I am positive on the ketogenic diet. I think it's hard, though, for adults to kind of do that. What we eat and meals are often great times of interaction and fellowship and time to be together. And so I know it's a struggle. I have a lot of patients who can do it, and I do think there's a role. On the natural product side, I think we got, increasing data that Boswellia, has a role, can help with controlling cerebral edema. The radiation oncologists are increasingly using that. I'm often asked about ivermectin and mebendazole, or the canine version for mebendazole. And, really the truth, ivermectin, it's a great antiparasitic drug, but within the field where I am, there's not a lot to support it as far as an anti-cancer effect. Mebendazole, there actually is a lot to support it as an anti-cancer effect. Dr. Greg Riggins, who's spoken here, and I know personally, was the initial one to describe this at Johns Hopkins. They've done clinical trials with this at Johns Hopkins. I think the challenge is able to get into a human levels into the nervous system that have an anti-glioma effect. All right, in summary, and just a few 3 slides left. So summary, got to increase our efforts to understand the environmental risk that's putting the right person in a bad situation. This is not like cancer in any other tissue. We must protect the nervous system. And then lastly, the immune system's role in the disease and the control of the disease is, in my opinion, the way forward. All right, I want to share with you some stuff because just like Dr. Musella and his team are being, advocates for May as Brain Cancer Awareness Month, we just finished 3 days of activities here in Birmingham. So Dr. Gelmelin, who's the co-leader of our program, we had the 3rd annual Southeastern Brain Tumor Research Symposium. We had about 8 institutions, about 100 people. It's graduate student led, so they kind of moderate the sessions, do most of the presentations. These are the award winners here in the middle. That's Anita. This is me on this end. We have a group we call Family Strong, which is our patient and caregiver meeting. We do this with the American Brain Tumor Association, so they're big supporters of that effort. We had a 4-hour meeting this past Friday with a number of speakers. We had some physical therapists in there, really giving them some information on activity at home. We had the arts and medicine folks, and then we had people from our palliative care and supportive oncology group. And then Saturday, as a massive thunderstorm rolled over Birmingham, it got by before we started. But I was out on getting the race set up at 5:30 when it kind of rolled over a place called Railroad Park here. We did the 12th annual Blazer Bolt for brain cancer. We had over 1,000 people register for this event, and I think most of them showed up. The rain held off. This is me. I did finish it. I ran the whole thing. I only stopped at the water break. And then this is the group headed off. And the last thing I like to sometimes share with folks is what I'm reading. A lot of my reading comes from books I find in the airport. I got this one, and I can't remember the name of it. I was in San Francisco for an advisory board at UCSF. name of it's Clear. It's a 19th set in 19th century Scotland. And it's about these what we'd call more poor type people, farmers usually that inhabited rural areas, and the wealthy landowners were clearing them from the land, and that's why the title Clear. Very interesting. I'm reading The Music of Bees by Eileen Garvin. I just got two beehives, so I'm an amateur, but I have two beehives in my backyard, thousands of bees in this thing. A patient, actually a family member of a patient, gave me a book, Capitalism in America, History. This is by Alan Greenspan, Adrian uh, Woodworth. This is a little slow. I have had no economic education in my life. So I'm finding this interested. I'm just reaching the part where the railroads are getting developed. I was reading a book called Rooted, which is by Leanda Alp, and it's called Life at the Crossroads of Science, Nature, and Spirituality. She references this book called Bless the Bird, so I ordered it by Susan Tweet. This is a special book. It is about the struggle with glioblastoma. Very impressive bit of writing, naturalist, recognizing a lot of stuff going on around them that I think too often we miss. And then lastly, I'm reading a book by Wendell Berry. He's an American writer, novelist. He's a farmer, big into agriculture. The Unsettling of America. This is about we're losing our healthy relationship with the earth, impact of large-scale agriculture on the planet and our health. And that goes back to some of this stuff we were talking about relative to diet and the immune system. So that is the end. Vanessa is back on, and Al.
I am back on, and we have, if you have time to stick around, we have quite a hodgepodge of
Yeah,
questions.
yeah, happy to.
My
So,
first is, how do you have time to do all this reading with all your professional roles?
most of my read, it's either on an airplane. So Clear is a little book I read, I bought, and so most of my books come from an airport. So I bought it and read it as I'm in San Francisco, get it Good Friday, and I get a text from Southwest, your flight is canceled. 1 o'clock Friday afternoon. Amazingly, got on another flight by Delta and got back early Saturday morning, so I could read that book. The Music of the Bees is very interesting. Capitalism in America is a slower slog. It's one of those that you feel like you got to read because you need to know some of this. And The Unsettling of America of Wendell Berry, I've read a lot of his nonfiction, which is an easy read. This is a little more laborious, but I think it's important. I think both of these books I think are important for helping us understand that relationship, between the economic development and the drivers, particularly within our country, and then where we get the majority of the food we eat, and then the food that, we largely are using to try to feed a big part of the world. The Bless the Birds I came upon by mistake and read it pretty quickly because it was in this other book I was reading, and she mentioned the author of this book. Her husband had died from a glioblastoma. She does a tremendous job. I would definitely read this book.
Mm.
Highly recommend that one.
Okay, so getting more into
Yeah. All
the
right.
topic of the webinar. So for that gamma delta T cell phase one you mentioned, is there a phase two? Is there any way to
Yeah.
ask?
This is a small biotech company out of Birmingham called N8 Bio. They had begun a phase two. Financially, it kind of got arrested. We're trying to reinvigorate that. We are going to amend the protocol for the phase one so that we can actually just add giving more cells more times. So I think we're looking at a way forward to keep that activity going. I am particularly interested in that kind of work, even to the point where we may do that before we did radiation. There was a time where we did neoadjuvant studies, which meant after surgery we did something and delayed radiation, and we kept close looks on patients. Back in that day, um, it was largely um, chemotherapy, poisons. It was not a biological mediator. So I think we'll get a cleaner way to look. So I do think those efforts will be going forward. They are going to go forward. I'm gonna, Vanessa, okay, here's one from Australia. Is that okay if I try to take this here?
Yeah, sure.
Uh, let's see. Yeah, so I mean, they kind of doing some of the stuff I just even talked about.
Oh, wait a second, Vanessa, could you just read the question so that people watching the video can?
Yeah,
Oh,
yeah.
okay. Here,
So and I'll moderate too, because there are a couple of related questions. So
okay.
aside from the gamma delta T cells, are there any other promising treatments out there? So the ones that people have mentioned so far are there's been a lot of questions recently since the peptide vaccine made in CGAT in Germany. We get a lot of patients ask about that.
Mm-hmm.
Richard Scolyer, world-renowned melanoma expert from Australia, he did a novel approach where he did some neoadjuvant, checkpoint inhibitors and then a combination of checkpoint inhibitors with a personalized vaccine. So those kinds of treatment strategies, do you think there's any hope there?
Well, I do. I mean, when you kind of think about it, and it becomes, it would like say in the United States to do some of this, it just gets getting the drug. I mean, I think a lot of people would do something that maybe is maybe it's a little more creative, maybe it's a little more preservation of the nervous system strategy. And obviously, I think, when we go back to kind of the epidemiology work suggesting an immune factor reducing risk. When we look at, our need to preserve the nervous system, a lot of what we do doesn't do that. And then lastly, as we get into what we're currently doing, we need to we need to deploy the immune modulating therapies earlier than later. Mostly we get stuck into doing stuff in recurrent disease, which often has been, depletion of the immune system. Patients' performance statuses are rough. Like all of this, I would like to see clinical trials and just, what we were just talking about, in this neoadjuvant window, meaning before, standard treatment. As far as peptide vaccines, I mean, that certainly makes sense. It's certainly shown benefit in other cancers. I think the biggest struggle, as I view a lot, is being able to get these, the vaccine that is generating an immune response for that immune response to find its way into the nervous system. That's kind of the problem in one big way. Um, here's a
On a related note, there's another question. So, what treatments or drugs do you find promising for modulating the immunosuppressive tumor microenvironment?
Well, I don't know that we have a ton of those right now, to be honest with you. So this work my lab's involved in, and fortunately, have somebody who I've worked with for 18 years. So Natalia Filipova, as we've been together that long, and is able to, keep the science going while I'm in clinic. But this inflammatory environment is not just promoting disease progression, it's damaging the nervous system. Okay. And so we definitely need to impact that. So, I don't know of drugs that are out there that would fall into the category of what I would call a TREM1 inhibitor or a DAP12 receptor, antagonist. But I do think not just in brain cancer, but even in other conditions where this is actually more recognized, like Alzheimer's disease, there are efforts to try to do that. Modulating the tumor microenvironment, and in the past we've used same strategy. So matrix metalloproteinase inhibitors, which are involved in the tumor microenvironment, we tried to use those, did not have any real benefit. So I do think we're going to have to extrapolate from potentially discoveries in other cancers, discover these ourselves. Obviously, all this, requires efforts for research, drug and therapy development, and deployment in such a way that we can confirm that these effects are occurring in patients or in patients we can select, we can pick out, meaning there's a biomarker, some way for us to know this is the right treatment for you, if that makes sense.
Absolutely. Um, Going through the questions, any particular thoughts on the Cervaxum vaccine developed out of Roswell Cancer Center?
No, no big
It's
thought
okay.
there. And and it's not that I don't agree. It's just I don't, I'm following about as much as I can follow.
Uh-huh.
So
Yep.
I do not know much about that.
Okay. And there's actually one I've never heard of. Do you know about astatine 211 alpha therapy?
Mm-mm.
I guess it did well with orthotopic tumors and improved survival
Well,
rates.
I will see, patients will often, which it helps me too, find some of the laboratory work. There is often a lot that is occurring in the lab for which we have a bigger path to getting it into a human acceptable molecule for dissemination. So we can do a lot in a mouse, and we actually cure a lot of mice.
Mhm.
And Braden's work kind of, I think, begins to illustrate why with a PD-1, we do great in mouse models of glioblastoma in a mouse microbiome. But when we implant, at least a human, then it becomes different. And that may speak, to a little bit of probiotics and, steroids and antibiotics are truly altering that, in our patients. I think we got to be cognizant, of that effect. But a lot of stuff that's in the discovery phase, it's a struggle, for us to get that from there into something that we can actually give a human.
Hmm. And what other clinical trials are you working on right now?
So we're working with the Ivy Foundation. So and so that's getting there, This is for
Hmm.
MGMT unmethylated, and so we're going to use a PARP inhibitor instead of temozolomide. It is a randomized study.
Okay.
So I think that is, trying to move things in a pathway. It's built on by the group out there with Dr.
Then yeah.
Sinai, strong preclinical work and previous publications. So we're excited about that. We have Agile open,
Okay. Yep.
and we just, I think we were the first site to get the new arm of an AstraZeneca drug. It's a
The
radiation
AZD
enhancer, AZ,
1390.
yeah, 1390. So we've got that open and screening patients right now. Um, we are amending the protocol for this INB. We have two oncolytic virus studies, and these are for newly diagnosed patients too, where
Okay.
at the time is Dr. Jim Markert's chair of neurosurgery. So at the time of surgery, and again, obviously you have to consent for this before, but you have implantation of a number of RICMs, which is an access device, kind of like an Omaya, but it's a little one. And then the virus is injected along with a PD-1 inhibitor longitudinally, even before you start chemotherapy and radiation, temozolomide and radiation. So
Hmm.
it's a lot of work for the patients and us, but I mean, I think it's helping us move this into the space we want it to be, which is more of the newly diagnosed patient. These are true pioneers that are helping us get these studies done.
Yeah, I agree with that strategy. And I know the NCCN guidelines are to consider clinical trials,
Yeah.
but there are a lot of patients who don't qualify. And for those
I,
later phases, even if they qualify, they can get into the control arm. So, how do you counsel patients on, you know, trying to enroll in a clinical trial versus maybe pursuing that personalized strategy of assuming you can get, you know, checkpoint inhibitors or vaccines or other things.
Right. That it's tough. And I think part of, I'm in the middle of kind of the deep South. And so people are coming from the Florida Panhandle and Mississippi. And, granted, that could be 4 to 6 hours, but you get to where you have to do that every week. That's a lot of logistics and time and energy for our patients and their families and caregivers. So I think you got to really, have kind of that discussion. Clinical trials, it's not like being on regular treatment. It's a lot more work for patients and their families, and I think that's underappreciated, particularly when we're dealing with a cancer like glioblastoma. It's a
Sure.
lot more. So I think we try to make sure people are informed, aware of that.
Okay.
Obviously required if we're going to make progress. There are certainly, options, but I will say there are not enough options. I think in, the close to 28 years I've been doing this, this is the fewest number of clinical trials since we really got going doing clinical trials that we've had in the portfolio. And right now, almost half of them are just investigator-initiated trials that we are creating here locally because there's a limited amount coming from, our NCI cooperative or consortia. And the consortia, the ABTC, the American Brain Tumor Association, the American Adult Brain Tumor Consortium doesn't even exist anymore.
Mm-hmm.
So I think a lot of that has put us in a position where we're, having to try to create more, clinical trials locally. Now, I do think we are more uh, taking advantage of advances in next generation sequencing, immune profiling for patients, trying to get that information, generate, a treatment plan that we may have to call off-label because it's not FDA approved. I think that's okay.
Mm-hmm.
I think particularly when you can identify a target which is altered in that patient, altered in the majority of tumor cells, that to me is a viable option. And we certainly try to take advantage of that. We're fortunate though. I mean, we've got a social worker who helps us. We've got a research on an oncology pharmacist who helps us,
Mm-hmm.
get this. And if you are out in community-based practice, you probably don't have resources to all that. Be very challenging to get an off-label, battle through a denial, an appeal, and then a patient assistance
Mm-hmm.
program application.
Yeah, yeah. The practical realities are tough. Do you have any thoughts on using organoids to test potential therapies in real time?
Yeah, so there are a couple of commercial platforms that have explored this. Um, We have active, research efforts here at UAB. Dr. Chris Willey, who's one of the radiation oncologists, has been, along with Dr. Gelmelin, have really, kind of worked this. The challenge is getting this real-time aspect, of it. I think there's information to be gained there, but we do probably have a time that I think we're often, feeling like we're a bit under the pressure, but I'm certainly supportive, particularly, obviously these are the patient's organoids. So it truly is looking for a biomarker that maybe is unique, to them. And I would
Mm-hmm.
search much rather if I found such information came back to me, use that even if it's not something, that we would lay down within our list of these are the common drugs used for glioblastoma, if we had some evidence that for that particular patient it would have been of benefit.
Mm-hmm. Okay. Can gamma knife be done before dendritic-based vaccines to increase its efficiency, especially
Well,
for unmethylated patients?
so, I don't know the answer to that. I mean, and we've certainly done that, though. We've certainly used, forms of focal radiation to enhance a downstream therapeutic, be that an immune checkpoint. We've done it with oncolytic viruses. So I think that's a very valid observation. I think it's supported by preclinical research, and there are even a lot of clinical trials that would be supportive.
Okay. And do you recommend, I guess this gets back to immune modulators, but do you recommend the tetanus vaccine to stimulate the immune system?
Well, this is kind of interesting, isn't it? That's the herpes virus we're using because everybody's got some degree of immunity to at least some version of the herpes virus. Polio was done at Duke, mumps at Mayo. I mean, we're trying to, it's not just the oncolytic capacity of the virus, it's the oncolytic capacity with an immune
Yeah.
event that we need. Since these are usually deployed straight into the tumor, then it's being created in the tumor's microenvironment. Um, I guess for tetanus, though, it seems like, I mean, in general, that might enhance, kind of a overall immune state. I think this is kind of also going back to these observations of Dr. Ostrom and Bontley where, people who have, some alteration, atopic dermatitis, atopic disease, autoimmune, so their
Mhm.
immune systems are in a heightened state, you know.
Mhm.
So, you know,
Okay.
wouldn't doesn't sound, bad. Uh, uh, so I think I could support that.
Okay. Is there any worry about developing viral-based therapies since we had the COVID pandemic?
Well, so, you know, the, obviously, I think one of the things of concern is there research ongoing where they were using gain of function to a virus. I will say as far as I'm aware of, most of the oncolytic viruses, it's really taking away some function. Okay. Example is herpes. Well, herpes you can't use in the wild type because it will cause herpes encephalitis that can kill you. So you have to remove genes from it, and the main genes that are getting removed are the ones for neurotrophism, making them want to infect a neuron. I think we always have to be aware, every time we learn something from an experience, I think we need to try to be aware of that. I don't think in oncolytic virus work relative to the current state for brain cancer work that there's anything there going on that we have to worry about.
Okay. And Darren asks, what about targeted therapies for PTEN deficient or RB1 deficient tumors? And he also mentions some authors were suggesting a synthetic lethality combination of niraparib and indotican for PTEN deficient tumors.
Well, and that's a common, combination of molecular alterations in tumors, RB alterations, PTEN, similar signaling. I don't know that we can, and we've had clinical trials. You can't target PTEN because it's a tumor suppressor. It's gone. You can't get it back. But when it's gone, then other parts of the cascade are activated. And so we've used inhibitors of those that have largely had unacceptable side effects or haven't shown any kind of benefit. Certainly, that makes scientific sense, but I just don't know that we can truly do that in a person very well.
Okay, we're almost through them all. I'll try to wrap up
That's
the
all
last
right.
few. So for the keto diet, can you speak to glutamine inhibitors?
Yeah, so give me a minute. I'm gonna have to so let me see what glut glutamine inhibitors. I'm gonna hit natural. Uh, so it's listing, some things. So I don't know exactly how you can target glutamine. So I'm probably don't know that I'm going to be able to answer that question. It's a part of the way that cells exchange molecules, particularly astrocytes. Their normal function is to take in the metabolites of what is involved in neurotransmission, glutamate being one of them, which is broken down into glutamine.
Mhm.
Uh, and the cell surface transporters can be inhibited. Dr. Harry Sontheimer, who's I think at UVA now, had done some work when he was at UAB in this space with a specific cell surface transporter that he had identified a drug that blocked it. But in
Okay.
general, I'm not too sure I know of glutamine inhibitors. And my little Google search is only bringing up some natural products that I've never heard of.
Okay, fair enough. All right, I'll try to wrap it up to
That's
two last questions that are probably universally applicable to GBM patients. Okay, so Optune is FDA approved and included in the guidelines, but uptake remains relatively low. In your opinion, how should clinicians talk to their GBM patients about Optune?
so I mention it to everybody. And so we were a part of the EF14 study and I think EF19. But I think this is a lot of work for patients, even though Novacure has made efforts to reduce the fingerprint on the patient. Okay. They've made some advances, the new little transducer things, the new thing
Flex
that sticks to
series,
your
yeah.
head, all is steps in the right direction. Um, you know, I'm and actually Doctors Willie and Gemlin have a cell culture Optune thing that they have been working with. I think I'm a little perplexed with the biological mechanism. I don't know that I truly get that, but I do think there is something to a cell's response to a certain frequency. Okay.
Hmm.
So I mentioned this to all patients. There's some clinical trials that you cannot use Optune. And then if you're not going to do a clinical trial, then it's, certainly supportive of you using Optune. I think though, for a lot of patients though, a lot of times when you're in medicine, not, not in it, but when you're as a patient having to come to medicine, we are doing it to you. You know what I mean? Like radiation therapy, you go get it done, then you go home, you're done with it. Optune's kind of with you always, and it's you and your family that are responsible. And the data is clear. You got to use this for it to help you. And I think that's kind of one of the big mindset struggles for some patients. Um,
I'm smiling because my husband is an Optune user.
yeah.
He's
Well, men, I have more success with men.
yeah, yeah.
Women, it's a big deal to shave your head, but I've got a lot of women who, do it too. So uh, there is more at play here. Granted, the side effects, the toxicities are much different, less,
Hmm.
but
Hmm.
there is an emotional and there's a bit of a burden you got to be able to accept. You know what I mean?
On the flip side, too, though, it is somewhat empowering because it's a part of our care journey that we can control.
I agree. Like diet. I agree. I'm totally with you on that. I
And
agree. I agree. I think, uh, and that I definitely recognize from, just my wife and our health things we've had to go through. Even when you're, in medicine, sometimes you're kind of at the mercy of medicine.
yeah, yeah. And I will say, one of the biggest frustrations we face on the patient side was that Optune was exclusionary,
Yeah.
prevented
Yeah.
us from enrolling in trials. And so we always advocate really strongly to
Yeah, I
allow
would
for
keep that
patients.
up. I would keep that up. I mean, I think the NCI cooperative groups are good. I think Alliance will allow Optune. So
Hmm.
Jan
Hmm.
Sikaria, Eva Galanis, I think that they're, for the most part, positive there. So you know, I would stay the course with that effort.
Yep. Okay. I will close it out with just one last question that comes up a lot too. So you talked about neuroprotection as kind of one of the goals we have to
Yeah.
balance. So for patients with recurrent GBM, how do you see the role of reirradiation?
Yeah. So, I mean, we are doing that a lot. And like if it's today, we kind of got to do what we got to do, and the patients are going to have to make these tough decisions. Um, and the radiation oncologists I work with are extremely close colleagues. We have just a great working relationship. But, I think part of this will fall into, kind of where you're coming from. I'm a neurologist, so my training was through, and even before was basic neuroscience and clinical neuroscience. And so, really carry a concern about, you know, that crucial, little edge we stand on where what we do helps control the disease or it causes the patient more harm. And it's
Mhm.
very difficult to predict that. You don't really even know. It is amazing. Some people will do just fine. Um, I don't think we have invested enough energy in neuroprotection. I think when you kind of think about it, they've done a better job in metastatic disease. So, hippocampal sparing, Namenda, if you're going to get whole brain radiation for multiple metastatic lesions, than we've done in, primary brain cancers. So, I think often we have to make these decisions. We're making these recommendations, may have to do something that we know, may have a cost, but, we're trying to utilize that. And that's, this book, Bless the Birds, that is a lot. And the author much more eloquently was able to present the struggle in those decisions than I can, as an engineer and a scientist. It left me recognizing this exists.
Mhm. Mhm. Okay, well, we've gone well over the hour, and
That's
I just
okay.
want to thank you so much for giving us so much time on a Sunday evening. We
And I
really,
came into my office. I was just terrified. My home internet just, it'll freeze all the time. I don't know what it is. I've got AT&T Uverse gigabyte thing, but like, if I'm doing a Zoom at home, it's plus or minus if it'll hold up. So this is my office. So you guys got a chance to see it.
well,
Well,
we
thank you so
appreciate
much. That was fantastic.
Yeah,
I could just
absolutely. All
talk
right.
with you for hours
You
like
guys
this.
stay in touch.
Yes.
Enjoyed it, and I'll keep working on iterations of this. It got me going, and I need to flesh it out more.
Thank you so much.
You guys
Thank
take care.
you.
Have a
Good night,
good
everyone.
week.
Wait, Vanessa, just mentioned
Oh,
we have more webinars.
yeah, we've been doing a series throughout the month of May, and we have one more at the very end of May. I think it's May 29th with Dr. Nick Batanza on B7H3 CAR T, targeting CAR T therapy in pediatric brain cancer.
Oh, we have two others. We have one.
Well, there's one more that's not in May. There's one in June
We have
also.
one Wednesday, May 21st, which is this Wednesday. That's in partnership with Mount Sinai.
Okay. Dr. Germanos. Yep. We
Right.
have that.
Okay.
All
Just check
right.
the website for details. Thank
Does
you.
look
And
like
good
I got a
night,
lot
everybody.
going.